message, and I will submit the work order now, so you don't have to do it.
Oh, at least now it's saying that. For like 8 years, it said, please clean filter now.
Stop class and clean the filter. Alright.
Yes.
I was going to.
Yes. When it
That's why I
meets,
want my tubes.
when it actually meets the end of usable life, we'll use that in the water.
That's what I'm gonna have my tubes have to say.
No
It
more
says
animal
HC
feed.
in that one, so, of her, reusable life. Perfect. Not anatomy, that is.
It's always sunny episodes of doing skiing. But
Electronic goods.
I used that day.
No, I've never done this.
Really?
Really. I've occasionally put a slideshow or something up.
Jackson cares about our environment, he does everything
I
electronic.
know. Well, like,
It also helps me stay
KCU was
organized.
like,
Great
yeah, you're gonna be
feedback.
getting an iPad and taking notes on your iPad. And I was like, I better get better at that.
The pencil actually does help things quite a lot.
It does. If you know how to use it, right?
Well-
It'd be time efficient.
Well, they offered to give us one because we had iPads and you guys were getting the pencil. And then I saw students use it, I'm like, 'cause I wasn't gonna get it, because I don't generally get technology just for the sake of having technology. And then I saw some of my students using it, I'm like, I might actually have to do that, so I did.
I just don't know if I can pay things as well if I write them on the screen.
Versus paper.
I know, it's weird, but it's.
Actually, I think studies would show the act of writing is what's important. It doesn't matter whether you're doing it on something digital or not. You could, in theory, write it on something that is ephemeral. And the act of the mental processing required to write actually helps. So it could be, you could write like an etch-a-sketch, if an etch-a-sketch would where you and shake it up and erase it every time, and it would indicate you.
Perhaps.
If the etch-a-sketch had an Apple pencil instead of
You came up with this story.
oh my god, it's the most frustrating toy
Yeah.
on the
I figured
planet.
it out on Friday, I was meant to tell you. That was easy.
Is that what you were looking for?
That's great.
That's a good question.
I feel like this message at the bottom should be stenciled on my forehead every morning. This human is reaching the end of its usable life. What's that? Are there?
Just a cool game.
Sadly.
Yeah, we're good.
Especially do the.
All right. Oh, 26 minutes left, or 26 seconds left, or more so. Although everybody's here, might as well just start.
Nah, let's just chill.
Our test is Friday, right? We have a test?
That is what the syllabus
Yeah.
says.
Oh, okay, that's what the schedule says?
Yeah.
No, I guess we're having a test on Friday.
Can over what?
Stuff that we've covered so far.
Let's talk about Wednesday, Paul.
What's wrong, Kennedy?
Yeah, that's a good point.
Can we talk about Wednesday?
Yeah.
Yeah, I'm for that.
Well, what do you want to talk about?
Can we go to the parade?
You may do whatever you want to, but if everyone appears to agree and nobody's going to be here, I would just appreciate you telling me now so that I don't show up to class and nobody else does,
Raise
because
your
that would
hand
be
if you're
cruel.
going to the parade.
Then the rest of us will be here having class.
Alright.
You have 2 unexcused absences, this burns one of them, so be in class the rest of the semester, except for one other day when you take another unexcused absence.
And you'll just post the video online?
I always post the videos online.
Can we get the links to those?
Oh, did I not send that out yet?
Yeah.
Oh, it's my same YouTube channel as before, but there's a playlist now, but I'll send you a link to the playlist. Yeah, Kennedy asked about this last week. Oh, and by the way, so I did not watch the Super Bowl, I'm not a football fan, but, there came a point during the evening where water pressure dropped at my house, just coming out of the faucets. I'm like, that's odd. I'm like, I wonder if it's halftime. And so I went and turned on the TV to see where the game was, and the game was actually in the fourth quarter with like 6.5 minutes to go, and they were down 20-10. I'm like, oh, that explains the drop in water pressure. They're going to lose the Super Bowl, and everybody's like been holding it in for the last 3 hours, and they're finally going to the bathroom. And then I started hearing like fireworks and horns honking and things like this. I'm like, what up? And then I turned on, I went and showered. So I was downstairs, then I went and showered because I'd gone on a bike ride earlier in the late afternoon, early evening. Went and showered and came back down, and then I heard the honking and the fireworks and everything. I'm like, what is going on? I turned the TV back on, and it was now whatever the final score was. Yeah, I'm like, what the hell happened while I was in the shower? Anyway, Kennedy asked me about this last week, and I basically said to her what I'm saying to you. You're free to do whatever you want. Just know that there are 2 unexcused absences, and this will be one of them. And just don't willy-nilly absences in the future, and have fun. I mean, part of being in college is learning how to balance life and work. And if it makes you happy to go to the Chiefs parade, by all means go to the Chiefs parade. Just know that those of us who are not going will be here having class. Other questions? Yes?
If there's a test on Friday, do you, like for this class, give us some sort of like guidelines, study guide?
Actually, in semesters past, I made up a list of the kind of sample questions that you might see on a test. That's in my folder. I'll put that on Google so you have that available to you. Because of the way that I have this course structured, and this is in the syllabus, so you can go and read the syllabus and see that it's also written there. So if I do something different, you can go to the syllabus and say, you said you were going to do X and you did Y. I concentrate on factual knowledge, so the tests deal with lower levels of Bloom's taxonomy. Do you know things? Can you remember things? Not. Can you analyze and apply and do a bunch of complicated things? As a result, it's a more straightforward test than perhaps what you're used to seeing from me, in that there are multiple choice tests, which I find harder to write because there's only one correct answer, and I'm much better at thinking about questions that have multiple correct answers, because I've got like 2 decades of writing tests that have multiple correct answers. So it'll be multiple choice and short answer, but
Multiple choice, or just?
no multiple choice. It's written in the syllabus. Did you read the syllabus?
I did not carefully analyze the syllabus.
And short answers, but the short answers will not rely on analysis, synthesis, creation, those higher levels of Bloom's taxonomy. And if you don't remember what those levels of Bloom's taxonomy are, they are also in the syllabus.
I'm just gonna go out on a limb and say, I'm assuming the literature papers that we read for class, fair game on the test?
Oh, yeah, sure.
Some teachers on the first exam, they will ask you questions about the syllabus. Is that something you do, or?
Yeah.
Okay.
If you care about what's in the syllabus, you should read the syllabus, and as a course that is being graded with specifications grading, you should care about what's in the syllabus. Logan, did you have your hand up?
I did, but I figured it out.
Claire, is your hand up or just your elbow? Yeah.
Are we going out tomorrow in the field?
Oh, yeah, good. I meant to say this. They're calling for sleet and snow tomorrow morning. And so we will meet, and it'll be like 26 degrees for a high tomorrow. And we will meet at 7:45 in lab, come dressed for indoor activities. We're gonna move up the Holling disk equation lab that was supposed to happen, not this week, but next week. And we'll put off goose observations until, the Holling disk equation lab takes us 3 weeks to get through. And so we'll just put off goose observations until better weather, hopefully, maybe, I don't know. I mean, we should have gone and watched geese yesterday out a Sunday instead of watching the Super Bowl because yesterday was great.
Today's great, yeah.
Today's okay, not nearly as nice as yesterday was, but
6 up.
we don't have enough time to watch these right now. Other questions? All right, we're talking about natural selection, and we had been going through the 4 forces of evolutionary change. The last of these forces is selection, and so when we think in animal behavior about the kinds of things that organisms have in terms of phenotype, behavioral phenotypes are seldom, either or kinds of phenotypes that we experience in like simple Mendelian genetics. When we think of peas can be tall or they can be short. They can have yellow seed coat color or green seed coat color. Their pods can be inflated or constricted. But when we think of behavioral phenotypes, oftentimes these phenotypes are more continuous in nature. How you forage, how much food you get per unit time, what your search method is, et cetera, those aren't these either or kinds of things. And so if those traits are heritable, they fall into the category of characteristics that some people refer to as complex characters and some people refer to quantitative characters. I call them quantitative characters because they can be analyzed with quantitative means, statistical means, and these quantitative characters are dealt with in genetics in a subfield of genetics called quantitative genetics. And if you remember this from evolution and ecology, a quantitative trait or a complex trait, depending on who you talk to, these are traits where each trait is undergirded by a number of genes where each gene contributes a small amount to that phenotype. Height is an example in humans, eye color is another example in humans, skin color is yet another example in humans. These are all traits that are determined by multiple genes acting on the same trait, and each dominant allele contributes something to the phenotype. These genes are inherited in Mendelian fashion, just like all genes are inherited in Mendelian fashion. Their inheritance isn't what's different about them. The action by which the gene gets expressed into phenotype is the difference. Each gene contributes a little bit to the overall phenotype. For these kinds of traits, you can calculate heritability using parent-offspring regressions. Heritability is determined with a statistical technique, regression. The slope of the line is heritability. Selection can be measured by looking at the phenotype of a population before selection and after selection. And if you have a heritable trait that is subject to some selection, it results in a change in phenotype in the next generation. I have traditionally written it this way. Dr. Reynolds writes it with a big R. The big R is nice because this is basically saying that the evolutionary response to selection is due to heritability multiplied times the strength of selection. And this is for directional selection in this case. This was all proposed by a guy named Ronald Fisher, R.A. Fisher, who was a geneticist and statistician who lived back in the 1930s, which is when this work was originally published.
What does R stand for again?
Response to selection. I always refer to it as the evolutionary response to selection because it's the response across generations. So if selection acts on a parental population during one generation, that heritability will carry that selection to the next generation and their response in the offspring. And this is why parent-offspring regression is valuable in looking at this. If you look at the phenotype of the parents and the phenotype of the offspring, it's this relationship that ultimately matters. These are some data that I got from what was the largest entering class of first-year biology students in the time that I've been here, we had 64 students one fall. That was interesting, and a ton of work. But we asked them to go home and get their parents, or call their parents and get their parents' heights in inches, and then to get the height of all their adult siblings. If they have little siblings, do not get them because they haven't finished growing yet. So adult height of parents and adult height of offspring, it would say mid-parent height down here at the bottom if they weren't covered by the wording about the lamp about to die. And what you can see here is that there's a lot of variation around the line, but the slope of the line is 0.9165. This is the y-intercept, and it basically is a relationship that describes if you have parents that are an average height, they will produce offspring that are almost that same height. The slope is less than 1. Heritability, h squared, can range from 0 to 1, 0 not heritable at all, to a slope of 1, which is perfectly heritable. In theory, you shouldn't be able to have a heritability greater than 1. But it's basically just showing you how parental phenotype gets translated into offspring phenotype. And in this case, it's pretty high. This is actually on the high side for height. So this is the heritability in this particular data set. Yeah.
So, is h squared always the slope of the regression line?
It is always the slope of the regression line when you are doing a mid-parent offspring regression. If you're doing something other than that, it's different. And there are books that show you how to alter this if you, for example, only have the mother and the average offspring, et cetera, et cetera.
What do you mean by mid-parent?
You have two parents, one parent's one height, the other parent's other height. You average them together, it's actually going to be the midpoint between the two of them. So it's also the average, but since there's only two of them, presumably, it's also the mid-parents, the midpoint between them. Average parent phenotype would also be suitable. So, what this means ultimately is if a selective event comes along, and in this case I've recolored the dots so that anybody who is below average height gets selected, sorry Haley, gets selected against. Are you familiar with that song from the 70s, Short People? No, it's a terrible song. It's very discriminatory against short people. If something happens such that if you're below average height, you can't reproduce either because you were killed off in some short person genocide, or just nobody wanted you, or whatever it was. You got killed by predators because you have short legs and can't run away. Whatever the situation is, this was the average height of the parental population before selection. So if this population were going to breed and have offspring, you could expect the average offspring height to be this value. But because we've selected against them, the height of the parents has shifted up because all those short individuals are gone, so the average parental height is higher, and because the average parental height is higher, the average offspring height that you would expect out of that population is also higher. This change in the phenotype in the parental generation is the strength of selection, S. The evolutionary response to selection is delta Z or R, and the degree to which selection influences the offspring is determined by what the slope of this relationship is. If the slope is steeper, selection is more similar to change in offspring. If the slope is shallow, more selection equals, the same amount of selection rather, equals less change in the offspring. And so the evolutionary response to selection is mediated by heritability and how that selection gets translated into change in the offspring generation. So this is where Fisher's equation comes from.
Can you say that part part about the steepness of the slope one more time in a different way?
Yeah, so imagine if this slope were not so steep. Well, I can actually draw it because I have this ability, because years ago I pestered the crap out of facilities. So this is the pre-selection mean of both parents and offspring. And so now imagine that rather than this slope, we have a relationship that has a shallower slope. The same degree of selection would only result in this amount of evolutionary response to selection. Selection is the same, heritability is lower, so the evolutionary response to selection is lower. Does that help?
Yes.
Okay. Oh, it's already there. So this is how it works. So for me, the easiest way to think about Fisher's equation in reality is to think about a parent-offspring regression, and as the individuals who make up that parent-offspring regression change, that changes the evolutionary response to selection. In this particular case, the difference between the pre-selection value and the post-selection value of parents was 2.41. Our heritability was 0.9165. And so the evolutionary response to selection is an increase in 2.209 inches. It's not quite 2.4 because the heritability is not perfect. It's not a 1, it's 0.91. And so you can select for almost 2.5 inches of selection on the parents, but that only translates into a little over 2 inches of change in the offspring population. Yeah.
How is the strength of selection determined? Is that just given or?
Dr. Reynolds was just talking about this equation before you guys all got here. So you can actually do this by measuring selection in the field. Who was I telling about Russ Landy and Steve Arnold? One of you was in my office.
Nico?
Hmm?
Galveston?
No, water bugs on the beach, Indiana sand dunes. Kenny. Steve Arnold, the book chapter that I asked you to read, Steve Arnold and his friend and colleague Russ Landy were both at the University of Chicago. While I was a graduate student, I was in charge of a graduate student speaker series, and we invited a lot of really kick-ass speakers to come and speak to the members of our department, because we had a ton of money in our graduate student bank account, because we rented lab kits to non-majors, which were like thousands and thousands of undergrads at the University of Texas Arlington. And if they rented them from us, the lab kit was in the lab and ready for them to use, and we kept them restocked, and you never got points off for not bringing your lab kit. And we rented them, I think, for $5 for the semester versus buying it at the bookstore for $15. And we got some bank, and we used that bank to invite in some of the best biologists that we could find. Steve Arnold was one of them, and I was the chair of the committee, and so I got to spend a lot of time with him at the time. And he told me the story of how the Landy and Arnold 1984 paper came about, where they talk about multivariate inheritance. And he basically said he was at a party at the Indiana sand dunes, Russ Landy was there, they were drinking, they were down by the beach on Lake Michigan. They saw a storm off on the horizon, lightning, thunderclouds, probably high winds. And after a while, they started noticing that these water bugs were washing up on the shore. And in their drunken haze, with all the beer bottles around them, they started noticing that some of the water bugs that were washing up onto the shore were alive, and a lot of the water bugs that were washing up onto the shore were dead. And they were like, aha, there's a selection event out over Lake Michigan, and it's downing the water bugs into the water. They're drowning and they're washing up on the shore. The ones that are dead clearly didn't make it and drowned. The ones that are alive clearly had something that separated them from the ones that were dead. So they ran up and down the beach, stuffing water bottles into water bugs into empty beer bottles. Took them back to the lab, measured their wing measurements, because wing loading is the thing in aerodynamics. Shorter, broader wings give you different flight characteristics than long, thin wings. And they measured the wing morphology as well as the body size, et cetera, and they examined this to see what selection was acting on in this moment. And so they had a population of dead bugs and they had a population of live bugs. Then they had a whole population of bugs. And so the difference in selection was the difference in the average wing morphology of all the bugs versus those bugs that survived. And so you can measure this directly when you see it happening. And they wrote a paper, and it was in evolution, and it was the first paper to actually mathematically model multivariate selection, and it came from hanging out on the beach and paying attention. So that's how you measure selection. You either see it happening, you stumble into it. In that paper, they talk about the Bumpus data set, which is a data set from the late 1800s, where a bunch of sparrows died in an unexpected cold snap, and Bumpus went out and collected the sparrows that were alive and the sparrows that were dead, and found differences in their morphology because their morphology had something to do with them being able to survive freezing to death during this unexpected cold snap. So you can measure selection directly in many cases. One of the things that Dr. Reynolds was talking about in terms of the flexibility of this equation is you can exert selection on a population of a known amount. You can look at the evolutionary response to selection, and then from that you can infer what the heritability of a particular trait is. So if you know heritability and selection, you can determine the evolutionary response. If you know selection and you know the response, you can infer heritability. If you, in theory, know heritability and the evolutionary response, you can infer what the strength of selection was that acted on that population in the past. Yeah.
Does selection have like heritability is 0 to 1? Like what is 2.41? Is that?
That's the average. That's the difference between the average phenotype before selection and the average phenotype after selection.
So is a larger number?
Yeah, a larger number. If we had killed off instead of, if rather than getting rid of these individuals here, we had come over to here, those would all be gone, which means that this mean would have moved over further. When this mean moves over further, that means the evolutionary response would have been larger because selection is stronger. So the evolutionary response to selection is larger when either selection is stronger or heritability is higher, or both. Weak selection, less response. Weak heritability, less response. Now, one of the things about selection is over-selection, as selection takes its toll on a population, what happens to heritability is ultimately heritability goes down. And so if you get rid of all those short individuals and look at the regression of what is left, you see that heritability now is down at 0.6615. So one of the conclusions of this is that selection erodes genetic variation. Selection gets rid of genetic variation. Fisher wrote heritability as h squared because this is a measure of genetic variance. This is called additive genetic variance because these genes essentially add on top of one another to create the phenotype. And because we notate variance as s squared, he made the heritability abbreviation h squared to remind people that this is a measure of variance. When you want heritability, you don't take the square root of h squared to get h. H squared is heritability. It's got a square because it's a type of variance. It took me 20 years to finally see somebody publish something where they told us what Fisher's rationale was for making it that notation. So this whole thing is heritability. It is this number. You do not take the square root of that. As a matter of fact, forget that square root symbol was ever even on the board. Okay? So don't do any mathematical machinations on the slope of the relationship. It's just the slope. Yeah.
So the h squared isn't like how likely it is that you're going to get a certain gene from your parents, or like, because to me, it's like if it's the same people, wouldn't you be just as likely to like get your height genes? Do you know what I'm saying? Like inherit height genes
I thought I was
regardless of
following you
like,
at the beginning of this question, now I'm lost a little bit.
like, you know, we could kill off my little brother who is taller than I am, but like, wouldn't my parents still have the same likelihood of?
Well, we're not selecting against the offspring, we're selecting against parents.
Mm,
So it isn't that we kill off your little brother, it's that we kill off your parents rather than killing off some other parents. Not that we're gonna kill anybody.
right, of course.
Yeah, so it's the genes and
One
it's the selection happens in one generation. The effect of that selection is seen within that generation as selection, as the strength and direction of selection. But you don't see the evolutionary response to selection until those individuals that survive and reproduce actually survive and reproduce and produce offspring. Does that make sense? Okay, alright, good. Yeah, Jess?
Is additive genetic variance just a synonym for heritability?
Yes, actually it is, yeah. So heritability is like one measure of additive genetic variance, certainly. In this case, we're measuring it through a regression. There are other ways of measuring it. There's a book, first author is Michael Lynch, and I forget, Lynch and Walsh, I think the second author is Walsh. It's about this thick. It's an introduction to quantitative genetics, and what it basically is it's like 400 pages of breeding designs that allow you to measure things like heritability. So you can do this by looking at parent-offspring regressions, but you can also take a bunch of lab animals. So let's say that you have 20 females and 20 males. You can mate each female to multiple males, get multiple offspring cohorts out of that, and then you can see how much of the variation in each litter is accounted for by variation among the females, variation among the males versus the variation of the two in combination. And from those kinds of breeding designs, you can also estimate heritability as well. Just depends on the kind of organism that you're looking for as to how easy, looking at whether it's easy to do that or it's easy to do a parent-offspring regression. If you're working on animals in the field, it's oftentimes easy enough to just collect the mothers, get their offspring, and do a single parent offspring regression because you can easily catch them in the field. In this case, you have no idea who the sire was, you only have the dam information, the female information. Once again, then that tells you not what heritability, but it gives you half of heritability because you only have one of the parents. And so there's lots of mathematical ways of getting a handle on what this is. Other questions?
Like, the breeder's equation really only works if you have inclined assumptions to something.
If you have what?
Like, if you're inclined to think this is gonna happen, then you can apply it. You can't just like, you know what I mean? Like height.
Well, I
You're
mean,
inclined to think that your height is from your parents, so you can use the breeder's equation.
you're not inclined to think that your height is a genetically determined trait that you inherited from your parents. Height is a genetically determined trait that you got from
Like,
your
just how, like,
parents.
I guess what I'm asking is just how practical is the breeder's equation?
So the breeder's equation was actually around informally before Fisher wrote it down mathematically because it was the way in which breeders thought about breeding cattle and domestic animals and things like this. So if a farmer, a dairy farmer, wanted to get more production of milk out of their stock, they would actually control the breeding of their stock such that only those cows that produce a lot of milk per day would be the ones that were able to breed, and over time they increased the milk production of the cows of their herd, which is why when you see dairy cows, you guys ever seen in Guernsey? Gigantic udders full of milk. That is the result of selection being enacted, conscious selection in this case being enacted by dairy farmers to increase milk production by controlling who bred and who didn't. So
But not because they use the breeder's equation.
no, they
That's what I'm getting at. You don't necessarily need to use a breeder's equation to get selection to act in.
no, but now you can say, but so now that you have it, you can say, well, I want a certain degree of change to happen in my herd from year to year. well, what is the strength of selection that I need to exert on that? Can I let, the top 15% of cattle breed, or do I just want one or two of my cattle to breed? Well, it depends on how fast you want that herd to increase. And as a result of that, you make different decisions. If you want fast change, you select for the particular traits that you are interested in, and maybe only one or two cows, let's say that you're breeding cows, have those traits, then those cattle are going to be very, very busy. And the rest of your cattle will not be bred at all. And so,
But then you'd be losing money by doing that.
pardon?
You'd be losing production by
Well-
not letting some breed.
Unless,
If you look at it that way,
well, it all depends on what your goal is.
yeah, that's true. That'll be more like a bull farm. You would select for a certain trait in a bull, something like that, or like a longhorn farm, something like that. Not like a,
You mean like longhorns?
yeah. Like registered
Double
Texas
helix
longhorn cattle would be,
ranch,
yeah, like I think you showed this in genetics, yeah.
Name their ranch Double Helix Ranch.
Well, this is the Double Helix Ranch because this is a ranch that is owned and run by David Hillis. David Hillis is a quantitative geneticist at the University of Texas at Austin. He is a phylogenetic systematist. He's a quantitative geneticist. He's published literally hundreds of papers about genetics, and this is kind of his pet project. So if one of your goals, and you can't appreciate this longhorn because this projector is at the end of its lamp life, but if your goal is to produce a grand champion Texas longhorn, then you control breeding because you want to get to that stage as quickly as possible, and you do it in such a way that's going to maximize your ability to do that. And so,
Purely for display.
but isn't it beautiful?
Yeah, but like what's the point of it?
That is a purdy looking longhorn. So, but it doesn't have to be purely for display. It can be things like,
Yeah,
how many pounds is the cow
yeah.
carrying on the hoof? How much drought resistance is this animal going to be capable of?
Yeah.
It can be milk production in the females. It can be any of these things that might be valuable to a breeder beyond just this cow looks purdy.
Yeah.
When you think about longhorns in terms of their evolutionary history is a breed that they were the descendants of cattle brought over here by the Spanish in the early history of the Spanish conquest of the New World. They got loose, they became feral, they went out into the wilds of southern Texas, which is very arid, and over time natural selection crafted this weird looking animal that's fairly lean, has these really long horns, doesn't lose a lot of weight when it roams around over wide ranges because it has to move around a lot in order to find good forage because it lives in semi-arid to arid parts of the desert Southwest. And when Anglo people came into the part of Texas where these animals lived, they found that they didn't need to bring their own cattle. They found these other cattle, if they just rounded them up, they could create herds of a group of cattle that basically natural selection had essentially adapted that population to that breed to the conditions that were found in that part of the world. Drought resistant, heat resistant, could keep their weight on very poor forage, et cetera, et cetera. And so when it came time to drive cattle from Texas to Kansas City, for example, during the time when we had cattle drives, you wanted something like longhorns that you could put through that journey and they wouldn't lose all of their weight on the way. All of those traits that I just described, the ability to keep your weight on the hoof, the ability to withstand drought conditions, the ability to withstand hot temperatures, all of those are quantitative traits. As such, if you wanted to select for those as a breeder, you could apply the breeder's equation and say, how much selection do I need to exert on my herd in order to get a given outcome? If you know the heritability, which you may or may not know. Hillis has been studying these, he's sequenced his herd. He knows a lot about these animals, and so he knows exactly what he needs to do to go from nothing to a grand champion longhorn in a relatively short period of time, which is what he did. Yeah.
Does the Brewer's equation apply to traits that are controlled by just a single gene?
No, it doesn't. Because those single genes are then going to have more either or kinds of effects. You're either this or that. It's not quantitative variation, it's binary variation at that point. We can still study them. They're still heritable in that they can be passed on from generation to generation. They're not heritability in that you can calculate quantitative heritability on them. Yeah.
You have a trait, like you were talking about, like ability to resist drought conditions. How do you make a trait like that quantitative? How do you measure it quantitatively? Do you just assign like a certain, like a 1 through 10 ranking, or do you have to get traits like how much urine production or something like
Yeah,
that?
so that, I wasn't thinking of urine production, but certainly that would be one thing that you can measure. So my partner studies copper resistance in fruit flies. So for something like cattle, it's a little more probably indirect. In Elizabeth's situation, she basically takes flies. She works with a group of strains where the flies in a particular line of flies are all genetically identical because they're recombinant inbred lines, rills. Are you guys familiar with rills? What you do is you basically take a single female, you put her in a vial, you allow her to lay eggs. After she's done laying eggs, you take her out, you leave those eggs in there. They develop into larvae, they develop into pupae, they develop into adults. You allow them to breed with one another. And then after that's done, you take that set of parents, you get rid of them, you take the next generation, you leave them in the vial, you let them develop, then they inbreed, and then they inbreed, and then they inbreed again, and you just keep this line from the descendants from this one female for about 25 generations, and in about 25 generations, about 98 to 99% of all of the alleles, all of the genes that you have are homozygous. So you can produce a true breeding population of flies just by exerting severe inbreeding on these individuals. So a guy at KU named Stuart McDonald did this with 1,700 lines of flies, and he actually put them together in a breeding design before he then created the recombinant inbred lines, and then he sequenced all of them. So he knows what the actual full genome is for all 1,700 of those lines, and because you know what their genome is, you can then take those lines and, for example, feed them on regular food or feed them on regular food that you've put copper sulfate into. And then what you do is you essentially see how long it takes for you to reach 50% death. And when you look at this, because I have to look at this a lot because she always wants to talk about work when she comes home, and so I have to get subjected to this. You see that the plot of death, you start off with 100% of your flies, and the ones that are on regular food, they're like, ooh, we're so happy, we're so happy, we're so happy. And then there comes a point where old age starts to set in, and then you finally start losing flies to old age. And this takes forever, like weeks and weeks and weeks. You'd be amazed at how long fruit flies live when you give them yummy food and a nice growing environment, et cetera, et cetera. Give them nice food, a great growing environment, et cetera, but then spike it with copper sulfate. This is what they do. Woo. Copper is toxic. And so you can study copper resistance by basically then asking, so at 50%, what's the average time to 50%? What's the average time to 50%? Under copper stress versus no copper stress. And then individual lines also respond differently. So some lines are copper resistant lines, some lines are not very copper resistant, but this is how you phenotype them.
But how does that true if they've all been inbred and they all have the same?
Well, they're all inbred differently, because each individual female was different. And so you sampled 1,700 females, and each of those is their own inbred line. And so the variations that those lines have are random, but for all the variations that they do have, they're homozygous, so they're true breeding. And so so there would be, if she did, so she's done this on the full panel, and so there are actually a bunch of lines here because they have different lifespans depending, and there are a bunch of lines here. Each line is the line-specific mortality, and so of these, some of these lines are more resistant than others of them because they lasted longer under copper stress than these lines did under copper stress. So whenever you're doing this, you have to phenotype your organisms for whatever it is. If you're investing in cattle, you wouldn't want to just put your cattle out there in the world and not give them any water and see who dies first. Although at the end of that, all the ones that died, you don't get to breed because they're dead, and the ones that did live, you would then keep them around because they're the ones that are clearly drought resistant. But you would maybe find some other way of determining this in cattle. You might hire a vet who comes out and measures renal function or something like that under drought stress conditions or something like that. But there are ways to phenotype organisms. If you're working with fruit flies, you can just see how long it takes them to die. If you're working with cattle, that's maybe not the phenotype that you want to measure.
Disposition to behavior.
Kennedy, was I talking to you about this, the foxes? Or was it, I was talking to you about this?
I don't think the foxes.
Who wanted to talk about dogs in their introduction? Claire, I talked to you about the foxes. Thank you for going away from that route in your introduction. There's this experiment. I did not see any reference to dogs in the thing that we looked at earlier today. The Russian fox experiment. This is an experiment that took place in Russia. It's been going on for what feels like forever now. They basically got a bunch of foxes, put them in kennels in Russia, and every generation they went up to the foxes, and if the fox snarled and bit at you, it was not allowed to breed. And if the fox didn't do that, they let it breed. And so they basically selected this population on the basis of one trait, essentially amiability, temperament, whatever you want to call it. They based it on, are you trying to bite me or are you not trying to bite me? And over the years, because they only let the nice foxes breed, the foxes got more and more tameable over time. Weird thing is, as they got more and more tameable, all sorts of other things about their phenotype changed. They got floppy ears, things happened to their behavior, like all of these other things that you wouldn't have expected to go along with this temperament trait that was the trait that was being artificially selected on. All sorts of their morphology even changed. I think the length of the snout changed, the size of the eyes relative to the head changed. All these other things changed. They were basically showing kind of what happened to dogs as they got domesticated. But so many of our dog breeds now have floppy ears, and people clip their ears to make them point out and stuff like that. And it's because the tameness, whenever genes control tameness, are linked to genes that control other aspects of the phenotype of the dog as well. How did I get onto this? Somebody asked a question.
Disposition.
You talked about this.
Oh, yeah, temperament, yeah. So, once again, temperament is a quantitative trait. It's probably a trait that is undergirded by multiple genes. We don't need to know what those genes are, we just need to know that there's probably more than a couple of them, and they lead to this complex additive trait that probably is normally distributed from really mean dogs to really kind dogs and a big bunch of dogs in the middle. And over time, we get rid of the mean dogs, we keep the nice dogs, and over time the population gets nicer and nicer, and as it gets nicer and nicer, other things also change in those dogs. Okay. Any other obscure genetic knowledge that you want to know about. What?
So the double helix ranch was good enough for me.
The double helix ranch is good enough for you. Selection erodes genetic variability. If selection acts long enough and is strong enough, it will basically wipe out all genetic variation, and then that trait will be fixed. You have 2 arms and 2 legs. That trait is fixed. You have eyes on the front of your face rather than the top of your head. That trait is fixed. Lots of your traits are fixed. Some of your traits are variable. Selection will ultimately drive to the fixation if selection is allowed to act long enough and if selection is strong enough. What Landy and Arnold did in 1983 is they basically expanded this equation to include more than one trait. So it's one thing to talk about one trait changing over time, but you and all organisms are a combination of lots of different traits. And as we saw in the fox example, selection against one trait, or selection on one trait, can influence the evolutionary pattern of other traits. And this is because selection on trait 1, which is beta 1, this is a selection gradient now because it's selection acting on that trait irrespective of any selection acting on any other traits that might influence trait 1. So selection on trait 1 multiplied times the heritability or additive genetic variance of trait 1 will change trait 1 over time, but selection acting on trait 2 can also affect trait 1 if trait 1 and 2 are genetically coupled, if there's a genetic correlation or covariance between trait 1 and trait 2. So if there's a genetic covariance, the change, the evolutionary response to selection is due to additive genetic variance times selection of trait 1, but additive genetic covariance multiplied times selection on trait 2 because of this covariance between trait 1 and trait 2. Yeah.
What is the difference between variance and covariance?
Variance is, so if you're going to think of it in terms of estimation, here's the easiest way, I think, to think about it. You would measure genetic covariance by taking so you would do a correlation between the offspring phenotype of trait 1 and the mid-parent value of trait 1. To measure the covariance, we would take the average offspring phenotype of trait 2 and see how it's related to parental trait 1.
So
So
the
if
variance
we-
is about one gene and the covariance is about,
So if this is the average phenotype of parents, we should expect the average phenotype of trait 2 to be this. If we select on parents on trait 1, if there's a significant covariance, it will change offspring phenotype of trait 2. Works the same way, except now you're talking about 2 traits rather than just 1.
so the G's with the same numbers are variance and the G's with different numbers are covariance.
And these 2 are the same value, because the genetic covariance between trait 1 and trait 2 is the same as the genetic covariance of trait 2 and trait 1. And there are some statistical niceties that we could go through and get into the weeds more, but this is essentially the idea. Yes.
Sorry, I missed you just now explaining what the G was, so 11 pairs with 21 somehow?
1, 1, this is trait
Okay.
1 to trait 1, so it's correlation between trait 1 and trait 1, this is correlation between trait 1 and trait 2, essentially. So the subscripts matter in this case. And so if you want to know what happens to 1, 1, 1 times 1, 1, 2 times 2. If you want to know what happens to 2, 2 times 2, 1, 2 is 2, 1 times 2, and 1, 2 times 1.
Why not two?
So wait, sorry. 2, 1 times 2 and 2, 2 times 2. No. 2, 1 times 1, 2, 2 times 2. It's easier for me to just write it
Mixed
out.
math is hard.
Actually.
Is that the rule, you like multiply the row by the column or something?
You multiply row times column, yes.
Okay.
See, I can do this without even looking up there. There.
Yeah.
I don't know why I can write it from memory and I get confused when it's on the board, but that's it. So the take-home point of this is that the response to selection is accomplished by direct selection on the trait acting on the additive variance of that trait, plus indirect, or what Landy and Arnold refer to as correlational selection, where another trait acts on the first trait through the genetic covariance. That times that, plus that times that. That plus that times that, plus that times that. Which is what I've drawn on the board. Sorry about the mathematical confusion. Matrix algebra is a little hard, but this is the only matrix algebra we ever have to do. This is the hardest math we will. Well, that's actually not true. Never mind, forget that. Organic can run more calculated math, or more complicated math tomorrow in lab, actually. Speaking of which, tomorrow in lab, on the Moodle site, February 4th, which is tomorrow, there is a reading on there by a guy named C.S. Halling, and that paper will be the basis of what we're doing in lab for the next couple weeks, so give that a quick read overnight. It's not really that long, it's just boring. Because it's written in 1959 and they wrote papers differently in 1959. But it's about foraging, and so it should be kind of reversible. So this is the multivariate version of the breeder's equation. The more traits that you include, the more likely you are to detect relationships among traits, the ways in which traits are linked to one another genetically. And when that happens, if you just use the univariate equation, if you just use this equation, you might find that the selection that you see acting on trait 1 times the heritability of trait 1 ends up not being the evolutionary response to selection, because there's also selection acting on other traits that is dragging trait 1 along. And that becomes important when you start looking at organisms as a whole. Selection times heritability should always predict the results, the end result, the response to selection, but sometimes it doesn't, because you have selection acting on other traits that are genetically related to the trait that you are interested in looking at. This happens to be the case in snakes. This is an example that we've told you about briefly in evolution and ecology. Little Butch Brody was working on this species of snake. The species of snake is Thamnophis ordinoides. It lives in the Pacific Northwest. In the same meadow at his grandmother's house, you can find the striped version and the checkered version living side by side. They're all the same species, but they have different color patterns. And one of the questions that Brody had when he was in grad school is, how can you have snakes of different colorations living in the same meadow? Seems like natural selection should select for either a striped snake or a checkered snake, because certainly both of these can't have equal selective advantage. And so he collected a bunch of snakes. He scored their color pattern such that he could attach a number to how stripy you are and a number to how checkered you are. And he and his dad raced a bunch of these snakes down a racetrack and measured their speed. He also put them in a circular racetrack and chased them around and around and around and around to see what their endurance was. So he was looking at these physiological characteristics to see how physiological characteristics might be tied to color pattern. And what they noticed at first was when they were racing them down a linear track, the track was long enough that when they had the babies, the track was too long for a snake to make it all the way down. And so that's when they started measuring endurance. But then, when they got them in the circular track, some snakes were more likely to reverse themselves, to change direction and move the other way. So they would chase them around this way, counterclockwise, and then suddenly the snake would turn his head around and go back clockwise. And so they began scoring the tendency of individual snakes to reverse themselves. And what they found, what Little Butch found, this was his dissertation research, is that there was a genetic correlation or genetic covariance between your stripedness and your behavior, your color pattern and your behavior. Striped snakes tended to run in a straight line and tended to not reverse themselves very often. Checkered snakes tended to run for a short distance and turn around and go back the other direction, or just turn around and stop. And so he scored little baby snakes, and then he scored them for color pattern, scored them for speed, scored them for endurance, and scored them for their tendency to reverse themselves, set those snakes loose out in this meadow, and then came back the next year and looked to see who had survived, who they recaptured. And what he found was that striped snakes that go in a straight line tended to survive better than all the other snakes. Checkered snakes, low on the stripe scale, that were high on the reversal scale, tended to survive better than checkered snakes that went in a straight line, and those are all way better than striped snakes that tend to reverse themselves. Here's the reason why. Think about your own visual bias. What kinds of things do you encounter that are striped? I think of them as actually banded because it goes across
Roads.
the narrow axis of the animal rather than going down the long axis of the animal. I think of zebras as banded, not striped.
Roads.
Roads are striped. What on the road is sometimes striped?
The lines,
Hmm?
like the center lines,
Yeah, what else?
tire marks.
Yeah, I'm thinking not the road itself, but what is on the road.
Cars.
Cars.
Who drives a
Okay.
striped car?
Who drives a striped car? So I just, me and Elizabeth just bought a Prius because her Volkswagen Jetta sucked. So that, it needed like $10,000 worth of work on it. And we were like, for $10,000 we could just get a new car. So we got a 2019 Toyota Prius on Black Friday last fall. It's white, it's sleek, it's clean looking. Guess what it doesn't have on it?
Racing stripes.
It doesn't have racing stripes, because what do you put racing stripes on?
Not a Prius.
Not a Prius. Give me an example of a car you might put racing stripes
A
on.
race car.
A race car, what else?
Mustang.
A Mustang,
Camaro.
Corvette.
a Camaro. Hmm?
I'm just kidding, sports cars.
Yeah. You put racing stripes on sports cars, you do not put racing stripes on a Prius. Why do we put racing stripes on things like sports cars and race cars?
They don't.
Aesthetic too, looks nice.
It makes them look fast is what it makes them do. Why does it make them look fast? What is it about the vertebrate eye that makes something striped look fast?
Makes you look like more shape.
What does your eye do with lines typically?
Follows them.
It extends them, doesn't it? If you want to look like you've lost weight, you can wear horizontal stripes, which I would call bands, or vertical stripes, going down the leg. Vertical stripes. Because it tricks your eye into thinking that you're taller and thinner than you actually are. Because the eye continues those lines upward. Same thing with a snake. A snake going in a straight line, the vertebrate eye extends that line out in front of them and especially behind that animal. Such that when you go to grab that animal, you oftentimes will find yourself grabbing air because you're not actually going for the tail, you're going for the residual stripe that is left after the animal has already passed because the vertebrate eye extends that line backwards. When you're a checkered snake and you're moving fast, what happens to that checker pattern?
Blurring into the line.
It just blurs into this general solid colored snake. If that snake then stops suddenly, what is your eye doing with that line?
Still standing.
It's moving forward to see where that line is going. If the snake stops and reverses itself and then stays still, what happens to that checkered pattern?
Changes.
It changes. It goes from being obvious when it was this solid snake going through the grass to now it's checkered, it's breaking up the outline of the snake, it's now cryptic. Checkered snakes that reverse themselves and stop tend to survive better than checkered snakes that go in a straight line. Snakes that go in a straight line and don't stop tend to survive better than striped snakes that stop and turn around and wait. Because what does that line do? It makes you stand out against kind of a random background, whereas checkered makes you blend in a random background. So what he found was that, well, he found a couple of things. He found that selection acted towards a combination of traits. Striped snakes that go in a line and checkered snakes that reverse themselves tended to have higher survival than the other combinations of phenotype. Hold on just a minute. The other thing that he found was that when he looked at the genetic correlation between color pattern and behavior, he found that they were correlated with one another. Alexis?
How do you know that it's not just the color pattern and then like the snakes just learn the best method to avoid predation?
Because he tested this in the parents and he tested it in neonate offspring who didn't have any experience out in the field. This was all done in the lab. And so the snakes that they're doing this with is garter snakes. The garter snakes, they can collect the female. The female doesn't lay eggs, they give live birth. And so they had the females, they could tell that they were gravid because you can feel the babies developing inside the gut. You can palp a snake and feel, you can count actually how many offspring are in it by just running your thumb down the belly. And so they would collect gravid females, they would bring them into the lab at the University of Texas at Arlington. They would wait for them to give birth, and then after they gave birth, they raced the females down the track and scored them for color pattern, and then they raced all their babies down the track and scored them for color pattern. And so they could calculate what the heritability is for those traits, and if they were learned, that would have broken down the heritability rather than contributing to the heritability. So selection, in this case, shaped the genetic correlation between these two traits. We're at 33 after. There are some math problems. We're gonna actually take, these are the values of heritability of reversal. The genetic correlation I've set to 0, and this is the heritability of stripedness. These are their observed, oh no, these aren't observed, these are, these are observed selection differentials on reversals and stripedness. And we're going to go through next time and we're gonna calculate what would the outcome of selection be if there was no genetic covariance versus if there is a genetic covariance. And these are the actual values that are from Brody's paper that was published in 1993 or something like that for this particular example. And you're gonna see that when we add the genetic covariance, the outcome of selection is very different. The outcome of the evolutionary response to selection is very different. And those of us who are here on Wednesday will do that, those of us who are not here on Wednesday will not.
Do it online.
Yeah, so these are up on today's date, and they're also up on a date from last week. I don't think the slides have changed things, but they're both places.
You said you'll send us that link to your.
Yeah, I need to remember to do that, but yeah.
Do you want a hard copy of the lab reports?
I prefer hard copies of lab reports. They're just easier for me to grade.